Welcome, everyone. It's a pleasure to continue the Josep Carreras Distinguished Lectures. As you know, in the Institute, we have a growing interest in the intersection between aging and cancer. And this is related to because many hematological disorders, they increase as we age. And we have examples in chronic hematopoiesis and others. So it makes a lot of sense to invite here Professor Björn Schumacher. Björn Schumacher is full professor and director of the Institute for Genomic Stability and Aging and Disease in the University of Cologne in Germany. He received his PhD at the Max Planck Institute of Biochemistry in Munich and did a postdoctoral research as EMBO and Marie Curie Fellow at the Erasmus Medical Center in Rotterdam. He's the current president of the German Society of DNA Repair. So as you know, DNA repair damage affects aging a lot, and has served as president of the German Society of Aging Research. He has received several awards, among these the Eva Louis Kohler Research Prize, the Innovation Prize of the State of North Westphalia, the DFG Koselleck Award, and also an ERC grant. His interests are in the molecular mechanisms that acting through DNA damage and other pathways are involved in cancer and aging. And he used for this a lot the model of C. elegans. Also, he has worked in different other models in biology. His interests are to, at the end, to prevent aging-associated disorders. So it's a pleasure to have you here, Björn.
Thank you so much for this very kind introduction, and it's a great honor to be invited here for this lecture here today. And what I want to do is tell you about the role of genome stability, both in aging and inheritance, and I want to tell you also how these two things, aging and inheritance, are actually interwoven. You might be all aware that we are living in the midst of the demographic change. The demographic change has started in about 100, 150 years ago when human life expectancy increased, and it has doubled since then. The traditional life expectancy was mid-30s. Now it's around 80 years for born today. And this has resulted in a demographic change, and that is not only a phenomenon in Western Europe and North America, but it's a worldwide phenomenon where now the proportion of elderly in society increases, and our societies here will soon reach 30% of elderly above 65. While this long life expectancy is one of the greatest accomplishments of civilization, old age invariably comes with disease. With age, dementia with Alzheimer's disease most prevalent. Cardiovascular disease still is the #1 killer. Kidney failure, a major clinical problem. Type 2 diabetes is on the rise. Age-related macular degeneration affecting the life quality of the elder. And the single biggest risk factor for cancer, more than any genetic or environmental factors, is age. But what is more is that not only we're getting sick when we are old, but one disease of aging rarely comes alone. Instead, the real disease of aging is multimorbidity. Multiple of these chronic diseases occurring at the same time, and the susceptibility for that is increasing exponentially after the 5th decade of life. The majority of elderly, in fact, suffers from multimorbidity. Over half of the healthcare costs today are spent on the elderly, and we will have 2 billion elderly by 2050 in the world. The traditional approach to these disorders has been, as you're well aware of, specialized treatment. To improve the treatment of cancer, dedicated cancer research tries to understand the biology of the cancer, and as you know, we've gone through personalized medicine to precision medicine to always more and more precisely define the therapy. And this is also true for cardiovascular diseases, the dementia forms, and so on. But when you now think that the patients suffer from multiple disorders and there's this huge prevalence affecting 1/3 of our population, that means this is unattainable for future in society. Aging research instead is postulating that there's not only a correlation between aging and these diseases, but instead aging might actually be the root cause for all these diseases. And so instead of treating disease once it has occurred, we want to devise treatments that target aging and protect against age-related diseases. Clearly, this requires that we understand the mechanisms of aging, and this is what we are striving to achieve. Now, what are the fundamental drivers of aging? A particularly important role comes to the genome, to the DNA. Why is that? Because the genome encodes all information, all biological information that you need to build and maintain a cell, but it is inherently unstable. The human genome is constantly exposed to damaging events. 100,000 lesions occur every single day in every single one of your cells in your body for your entire life. They can come from the environment, they can come from endogenous metabolism, and they cause a plethora of different damage types. There are consequences of DNA damage. The most well-known consequences are mutations. We know for a very long time now that genetic aberrations can be caused by DNA damage, aneuploidy translocations, deletions, mutations, and this is known for many decades now. This is why we get cancer. Cancers caused by mutations. But DNA damage can also lead to blockage of transcription and replication, and this leads to signaling of the DNA damage to affect cell fate alterations. Cells might undergo apoptosis, they senesce, stem cell compartments get deprived, some cells undergo polyploidization. And these cell fate alterations then drive the aging process. The cell's functions decline, tissues become atrophic, and inflammation becomes systemic. DNA repair mechanisms have evolved early in evolutionary history because they are absolutely essential to prevent these detrimental outcomes. And here I show you what happens in humans that are born with DNA repair defects, and they really exemplify the role of DNA damage in both cancer and in aging. You see here two boys that phenotypically couldn't be any more different, yet their molecular dysfunction affecting nucleotide excision repair genes are very much interconnected. Here you see mutagenic consequences of a DNA repair defect, xeroderma pigmentosum, where already in childhood, multiple skin cancers are forming. There's several thousand-fold increased susceptibility. Normally in humans, skin cancer takes decades to evolve from the first UV damage. On the other side, you see a cocaine syndrome patient who already in the first decade of life shows initially severe growth retardation and then multiple signs, pathologies of premature aging, retinal degeneration, neurodegeneration, atherosclerosis, typical pathologies that we only get when we are very old. One defect in one DNA repair gene is sufficient for that. Now here we get a glimpse of mechanisms underlying aging and cancer development. But let's start with the ultimate question, why do we actually age? And that clearly requires that we understand the evolutionary history that has formed the aging process. And here the fundamental contribution came from a zoologist, August Weismann, who back in the 19th century was one of the three top evolutionary biologists after Darwin and Wallace. And what Weismann proposed was the germplasm theory, he called it back then. It's still today the current theory of inheritance. Weismann realized, and back then it wasn't trivial, today you all know it's true, but back then it was not trivial, that heritable traits are exclusively passed on by germ cells. Only germ cells perpetuate the genetic information. And they are actually protected by what was later known as the Weismann barrier from somatic and environmental influences. And when he realized that germ cells indefinitely pass on the genetic information throughout the generations, it occurred to him that indefinite maintenance of the soma would offer no benefit for the fitness of the species. So therefore, upon installation of the next generation, the soma can decay, age, and die. And that is fundamentally why we age. We are nothing more than the vehicles for our germ cells to pass on the genetic information. This also means that you have indefinite germline maintenance, and that must mean that also the genomes. We have essentially the same genomes as our ancestors a few 100,000 years ago. They've been maintained all the way through germ cells. While in the soma, there's only a single generation maintenance. So we propose that we can learn some fundamentally interesting things by studying the genome stability mechanisms of germ cells. Now, germ cells, however, are different, particularly in humans, but in most species. And there are sex differences that are important in what are the contributions to inheritance. The female germ cells provide most cytoplasmic components and organelles. Female germ cells are the predominant source of aneuploidy. And aneuploidy, trisomy 21, for example, the biggest risk factor for trisomy is maternal age. The paternal germ cells do something fundamentally different. They are the predominant source of mutations. 80% of single nucleotide but also structure variants come from the paternal germ cells. So therefore, there must be important differences here. What we decided to study in the lab are therefore the consequences of paternal DNA damage. And this has been really pioneered by an outstanding scientist, Xiang Wang, and the bioinformatics analysis I show you is from David Meyer in the lab. And the background of why these transgenerational effects of DNA damage are interesting is that we have in recent decades seen a huge increase in neurodevelopmental disorders. The autism rates are really increasing rapidly. And they have been linked to de novo paternal chromosomal structure variants. So there could be something linked to that. There's a long ongoing debate, and you probably know much more about this debate than I do, about whether there is an increased leukemia risk in children of radiation-exposed fathers. A lot of controversial literature on that. But recently, about 2 years ago, in the UK Genome Project, they found hypermutated germline genomes in humans. And these were very rare events that occurred, and they were associated with those fathers that were that had genotoxic influences. Like, for example, they underwent chemotherapy at a time close to conception. So there might be something to this. But instead of studying epidemiology, we thought we should seek the underlying mechanisms. And that's where we turn to our favorite model organism, C. elegans. And for those who are familiar with C. elegans, it might not occur immediately why you would use C. elegans to study sex differences when they are normally hermaphrodites. But we can take advantage of mutation in the sex determination pathway to generate separate females and males. And then we can study them. So what we did here, a very simple initial experiment. We took animals, adults, and we treated either the female with ionizing radiation and then mated it to healthy males. And these females we would call delta F, and then we scored the lethality of their progeny. And as you can appreciate here, in their progeny there's a high degree of lethality because they experience DNA damage, just as you expected. The survivors we took, either the daughters, the delta FF, or the sons, delta FM, and then we mated them again with healthy opposite sex. And then we scored the lethality in their progeny, and there was no lethality, as you would expect, because the ones that grew up there have repaired by then. Then we did the exact same experiments with males. We irradiated males, delta M, and scored the survival of the progeny, and we saw nearly everybody survived, despite the same dose of DNA damage inflicted. Then we took those animals, the daughters or the sons, and mated them with healthy opposite sex. And then something very unexpected happened. their progeny, here the daughters and the sons, showed an extraordinary high lethality in the next generation. And you can appreciate this is even more profound in the progeny of the daughters than of the sons. I can tell you later why that is. Then we ask, is it a specific stage, a specific type of cells during spermatogenesis that is accountable for this? And we did, again, a simple experiment. We did the same thing. We radiated a male, mated it with an unrated female, and we saw in the offspring of their in the offspring of the daughters and the sons, we saw again this very high lethality. But then when we let the worm, the male recover, the father recover, and mate again a day later, there was no such effect. And worms, just like humans, they produce, they have ongoing spermatogenesis all their life, and they store then the mature sperm until they ejaculate 70% during a mating. And that's what happened here. This was mature sperm, and this here was newly generated sperm. That obviously can repair. Does this also happen in hermaphrodites, the normal form of C. elegans? Here we use hermaphrodites, and hermaphrodites, they switch sex in their germline. They initially, during the larval stage, they produce a sperm, and then they switch and produce oocytes for the rest of their lives. So during the third larval stage, L3, and in the young L4s, there's ongoing spermatogenesis, and we see no such transgenerational effect here in the progeny of F1, F2, no such effect. But then when we irradiate the last larval stage, L4, where all the sperm is mature, then we see this very strong transgenerational lethality, and it maintains, because here they are reproduced by selfing, so there's no mating with healthy opposite sex. Then we ask, how long does this hold on in hermaphrodites where they interbreed with their own germ cells? And it essentially goes on forever. So the rare survivors, their progeny is again in every single generation has very high levels of lethality. Now let's look what happens to the genomes of these fathers. So we irradiate the male, and we look now at the chromatin structure of the mature sperm. Normally highly compacted chromatin structure, just like in human mature sperm, very, very compacted. And this is kind of dissolved a bit after ionizing radiation. So it's not so compacted anymore. Nonetheless, despite the damage, they fertilize an egg, and then these eggs, they form an embryo, and the embryo shows overt signs of genome instability, Darby bridges, micronuclei. Despite all this damage, despite all the genome instability, they grow up to adult worms. And now we look at the soma of the offspring, and we see here intestinal cells control, but here in intestinal cells, they are very large nuclei, so we can visualize that. They are Darby bridges, indicative of genome instability. Despite numerous cell divisions, still ongoing genome instability. Then we look at the germline now of a daughter of such an irradiated father. And what we see here is control, and here's a germline of the daughter. We see appearance of genome instability in germ cells. So they have already proliferated, they form these new meiotic cells, and we see micronuclei. In the oocytes, then we see overt signs again, fragments of chromosomes still in the daughter germlines. And that accounts then for the ensuing lethality in the offspring. Then we sequenced these genomes of the offspring. So we sequenced the father that experienced irradiation, the mother, the son, and the daughter. And we have many of those here. I show two examples of a daughter and of a son. And you see here the fusion events, the translocations all over the place, involves every chromosome and all that. There's one difference between those two, because only in the daughter, these fusions involve the X chromosome, because one X of the daughter comes from the father, while in the son, they only get the X chromosome from the mother, the X0, so it's only from the mother. That also already told us that these fusion events only occur within the paternal genome. They do not involve the maternal genome. And this also explains why there's so much more lethality in the in the offspring of the daughters, because when you have X fused to anything autosomal, then you cannot silence the X chromosome during meiosis, which gives you an extra lethality. When we look now at these fusion sites, we observe a microhomology of one nucleotide. And in C. elegans, a microhomology of one nucleotide is the signature fusion site for a repair mechanism that is called polymerase theta-mediated end joining, TMEJ, relatively only recently discovered repair mechanism. Because for double-strand breaks that we inflict by this ionizing radiation, there are three major repair mechanisms. There's homologous recombination repair, very accurate. There's non-homologous end joining, inaccurate. And there's this TMEJ with this microhomology, also inaccurate. Then we tested genetically which one was most contributing. And so we do this all always in FOC2 mutants. These are female-only germlines, so that we have males and females. That's just the background. But then we inactivate BRCA1 that abolishes homologous recombination repair, CQ70 that abolishes non-homologous end joining, and POLQ1, which is Pol theta, abolishes TMEJ. And what we see is that specifically in the TMEJ mutants, there's an exquisite sensitivity. So already in the offspring of the father, there's directly an extraordinary high lethality. The few worms that survive cannot even form a germline, so we can even look at them in the next generation. There's some contribution also of non-homologous end joining. You see this here, but no contribution at all by homologous recombination repair. They are never different to wild type. So despite being germ cells, they do not use homologous recombination repair. Then we ask who repairs, the father or the mother. So here again, we do the same experiment. Both father and mother have a Pol theta defect. And these are just controls. This is irradiated fathers. And we see that their offspring show this very high lethality. But when only the father is Pol theta TMEJ defective, there's no such effect, indicating that the father is not repairing. But when the mother, that actually never experienced the damage herself, is defective in Pol theta, we see this enormously high lethality, indicating that the repair is mediated by maternally provided TMEJ in the zygote. So there's no repair on the mature sperm. They are very highly compacted chromatin. There cannot be any repair. Once it fertilizes in the zygote, the repair happens by maternal TMEJ. Now you might argue, this is a lab, we use ionizing radiation. Does this ever occur in nature? So we looked at the genomes of 540 natural isolates of C. elegans all around the world. They've been apart for millions of years. And we asked those structure variants that they show in comparison to the lab strain, do they have the same signature? And so here we have the expected rate of this microhomology. In the natural variants, we see a very high enrichment for these microhomology fusion sites. They're highly significantly enriched. Then we ask whether in mutation accumulation strains, so these are like 40 generations of lab strains, where you always maintain a worm for another generation, and they naturally then just accumulate mutations. And also here we saw then they accumulate the TMEJ fusion sites. Now you might argue, I'm not a worm, I'm a human, is that relevant for me? And then we looked at human data genome sequences, the 1000 Genome Project and Polaris data set, and asked, do we also find in the structure variants a microhomology TMEJ type signature? In humans, in contrast to worms, the TMEJ signature is 2 to 6 nucleotides, while in worms it's predominantly 1 nucleotide. And we find again a strong overrepresentation of these TMEJ fusion site. And then we ask whether in humans, like in the worms, this would also occur in the paternal genome, in the father's germline. And that's where we use the Icelandic studies, where they sequenced trios, mother, father, child. And we saw only in the paternal, in the DNA of the fathers, an enormously overrepresentation of the TMEJ signature. This is shown here, but not in the mothers. So that suggests that this mechanism is conserved from worms to humans. Then we wanted to understand how this all works. Why is there transgenerational lethality? How can the genome, the unstable genomes be inherited? We did a mass spec experiment of offspring of irradiated animals. And in these offsprings, we found an enormous enrichment of protein DNA complex assembly. And when we, many of them are actually histones. And when we look at that, we find specifically in very strong induction of the H1 linker histone. It's very much highly expressed, these proteins. Then we asked, are they responsible for the transgenerational lethality? We here mutated one of the a viable subunit, a non-essential subunit of H1 linker his24. And indeed, in the offspring of the daughters and of the sons, we saw a reversal of the lethality in the offspring, suggesting that this induction of H1 linker is responsible. Now, what are high levels of H1 linker associated with? They're associated with heterochromatin. They increase the compaction of chromatin. And this is exactly what happens. So here we look at a heterochromatin marker. Here in the germ cells, this is H3K9 dimethylation, and we see a very strong increase in dimethylation levels in the germlines of the daughters as well as of the sons of irradiated fathers. Very strong high levels quantified here. But when we then knock down the H1 linker component his24 or the heterochromatin protein HBL1, we can reverse these high heterochromatin levels. Now, we ask what could be the consequences of this heterochromatization that could be reversed by removing the H1 linker levels. And that is access to accurate repair. So here we are now in the germlines of the daughters or of the sons. And despite this very high lethality that they give rise to in their offspring, they have relatively few foci of RAD51. RAD51 is a key component of homologous recombination repair. That's the normal repair mechanisms, highly accurate, that is used during gametogenesis. But when we then knock down his24 HBA1, then we see much higher levels of RAD51, suggesting when we now remove this increased heterochromatization, now homologous recombination repair gets access and can restore stability. Now we ask, lastly, whether the engagement of homologous recombination repair was the critical factor that allows the animals where we now alleviate heterochromatization to give rise to viable offspring. And this looks very busy, but let me walk you through this. What we have here are, again, the daughters and the sons of irradiated fathers. We see again this high level of their lethality of their progeny, both sexes. Then we see next that when we knock down his24 HBL1, we can alleviate this lethality, both cases. But then, if we then in addition eliminate BRCA1, which is the initiator of homologous recombination repair, the suppression vanishes again, indicating that it is the engagement of homologous recombination repair that can revert the genome instability in their germ cells. What did I show here? I showed you that there are consequences transgenerational when DNA damage occurs in mature sperm. These breaks cannot be accessed by any repair in the mature sperm. The chromatin is too compact. They only are repaired once they fertilize an egg by maternal theta-mediated end joining. This, however, results in fusions. It's very inaccurate repair mechanisms. These fusions then lead to ongoing generation of genome instability because, as Barbara McClintock showed in the 1940s, fusion events and translocations can give rise to the breakage fusion bridge cycle that in every cell division gives rise to new genome instability. This is what we believe is leading then to these high levels of genome instability in their daughters and their sons. In the germlines, this is reacted upon with strong heterochromatization. And this heterochromatization prevents access of homologous recombination repair, and that leads to high embryonic lethality. What did I show you here? We found that there's inheritance of paternal DNA damage by histone-mediated DNA repair restrictions. DNA damage that is inflicted in mature sperm leads to genome instability in the offspring and transgenerational lethality. The maternal TMEJ is the major repair mechanism for paternal DNA damage, but leads to chromosomal fusions. But the paternally inherited structural variants also in humans carry the very same signature of TMEJ. The H1 linker mediates heterochromatin formation and prevents access for homologous recombination repair in the offspring. And the transgenerational DNA damage can be repaired when we delete the H1 linker component or heterochromatin factor to then allow homologous recombination access of the inherited DNA damage. And this really provides the mechanistic underpinnings of germline hypermutation in mature sperm. Now, for the last bit, I would like to switch back to our thoughts at the very beginning when we thought about the distinctions between germline and the soma and how this is important in the indefinite maintenance of germline genomes while somatic repair clearly is restricted. And despite what I showed you about inheritance of mutations, and they clearly occur, with every year of male lifespan, there are 2 de novo mutations in the germ cells, so clearly there are mutations, but somatic mutation rates are by more than an order of magnitude higher than germline mutation rates. We asked the question, what we were interested in is whether we can increase DNA repair. Because a question that has really been unanswered for decades in the field is, how can we increase DNA repair capacities? I showed you that we can inactivate repair in these genetic syndromes and promote cancer, promote aging, but the opposite has not been accomplished. The problem here is that there are hundreds of distinct damage types, at least 6 major and additional systems that are required to remove all these distinct sites. And so far what has been accomplished is only that single enzyme repair mechanisms could be improved. So photolysis are an example of that. They repair the thymidine dimers that UV induces. We don't have these photolysis as humans. Plants have it, for example. We can use it in skin lotion, and then we can use these enzymes, but we don't have them inherently. And we asked the question whether they're master regulators of DNA repair. And where would we look for that? And as I already nudged you into this, a good place to look for that is what distinguishes somatic and germline repair. Because germline repair, despite all what I showed you before, is still much more accurate than human repair. And this, in worms, this is the same, but much more clearly defined, because there are some repair mechanisms, like homologous recombination repair, global genome nucleotide excision repair, that are only present in germ cells. The soma doesn't even bother to express those genes. So then an extremely talented grad student, Arturo Bujabel Dueso, asked the question whether he could find evidence for a master regulator that would determine the expression levels of DNA repair genes. And what Arturo did, he looked at all the DNA damage response genes in C. elegans, and C. elegans has the advantage that the promoter structure is very simple. It's not these complicated things mammals do. So they're relatively defined with a few kb upstream, and he looked for whether there was any transcription factor target motif overrepresented, and he found one very unique specific one, the CDE-CHR motif. And the CDE-CHR motif is the binding site for the so-called DREAM complex that binds to these elements and represses the target genes. And we didn't come up with the name DREAM, although this would not be inappropriate, but it stands actually for dimerization partner RB-like proteins E2F SMIF-B core. And our hypothesis was then, because these elements are repair genes, that the DREAM would then repress the repair genes. And there are many things known about the DREAM, particularly in C. elegans, and what is known is that the DREAM is actually present only in somatic cells, not in germ cells. So it was a pretty good place to start. So then, what we did here, we used L1 worms. These are the first larval stage of worms, They are 90% of these cells are already post-mitotic, and they then expand in size mostly to form adult animals within 48 hours. And that you can see here in the control. When we then apply DNA damage, in this case we use UV radiation, we then score how far have they gotten by the same time point. And we see here with 60 millijoules per square centimeter UV, for example, we see 40% of the wild-type worms are still L1s. The rest have gone to the L2 second larval stage. So they are severely delayed because of the DNA damage. Now we tested Lin52, DPL1, EFL1, Lin35 mutants in different subunits of the DREAM complex, and all of them show a significant advance in development compared to the control. So here everybody is still arrested. Here the animals are already at the third larval stages, even the high dose, indicating that they are resistant to DNA damage. This is a bona fide function of the DREAM complex because here we test Lin52 and DPL1, two different components, and they are epistatic because the double mutant has exactly the same resistance as the single mutants. So the DREAM function. That's development. What about adulthood? Now in adults, there's an important difference between C. elegans and humans. I showed you in humans, one defect in the DNA repair gene is sufficient to accelerate aging. In worms, they are too simple and too short-lived for that. We need to treat them with DNA damage. That's in the solid line upon DNA damage. We see a lifespan reduction that is significantly extended by all of these different mutants in DREAM. So they are longer lived after DNA damage. Do they repair better? UV lesions, 80% of the UV lesions are dimers of two adjacent thymidines, and these are called cyclobutyl pyrimidine dimers. And we have an antibody that binds to these CPDs, to this thymidine dimers. And after UV, we can detect them in the slot plot in the DNA, they're formed. And after 24 hours, there's some repair in the wild type, but there's much more efficient repair in Lin52 DPL1 mutants, indicating there's increased DNA repair. This also happens in adults. Here we see a head region of an adult. Most of these cells are neurons, and there's very ineffective repair in the wild type, but a much more pronounced repair in Lin52 mutants, indicating that really DNA repair kinetics are better. But I showed you that we find these DREAM targets sites in all types of DNA repair genes. So we then did an RNA-seq experiment of the Lin52 versus wild-type animals, and consistent with the Lin52 mutants taking out a repressor, we see an induction of many genes, and a large number of those are DNA repair genes. And what is more is that these repair genes that are now de-repressed in the DREAM mutant and the Lin52 mutants, they operate in nucleotide excision repair, interleukin cross-link repair, base excision repair, molecular recombination repair, mismatch repair, non-homologous end joining. That then let us hypothesize that if repair genes in all of these mechanisms are induced, do they become resistant to all sorts of DNA damage and not only UV lesions? And this is indeed happening. So here again, we use ionizing radiation. Here, this is in development, so they're post-mitotic cells. That's why we have to use very high irradiation doses, because post-mitotic cells are much less affected. We see here again a significant resistance of Lin52 versus wild type. This resistance dependent on non-homologous end joining. We inactivate non-homologous end joining. We have no such effect. Then we test alkylating lesions, MMS, and we see again a significant resistance. And we can suppress even single DNA repair defects. Here, Paulita, for example, that leads to a hypersensitivity to alkylating lesions, can be suppressed by Lin52 mutants because other repair mechanisms can take over. Also, cisplatin, frequently used still nowadays in chemotherapy, there's a significant resistance. Now I show you an experiment that we found particularly exciting. Here we now don't use these L1s that are mostly that are almost exclusively post-mitotic, but we use embryos, early embryos. They now will undergo the most rapid cell divisions of any stage of C. elegans to form these L1 worms. When we apply ionizing radiation, these embryos instead die. And we can see that in the solid lines, they die with increasing IR. ELIN52 mutants, however, can significantly induce resistance to ionizing radiation, even in this proliferating cell types. These early embryos, because they are very, very early, they use the homologous recombination repair for repairing, which we can completely inactivate by BRCA-BART1 double mutant. Then they are hypersensitive compared to wild type, right? But ELIN52 mutation is sufficient to revert their hypersensitivity because other repair mechanisms can take over. In this case, it's NHEJ takes over. When we do the reverse NHEJ inactivation, homologous recombination repair is more effective, takes over. Again, our worms, is it relevant for humans? The DREAM complex is highly conserved in humans. It's exactly the same protein subunits that make up the DREAM complex, exactly the same target sequence, CDH, CHR. And then we ask among the DREAM targets, they are validated with ChIP-seq of all of these different subunits of DREAM. Do they contain DNA repair genes? And there's indeed a significant enrichment in DNA repair genes in the directly regulated targets of the DREAM complex in humans. Now in humans, everything is more complex. Different subunits of DREAM can also function in other complexes, so you cannot just make a knockout or something like that. But there's also advantages in humans, and particular advantages that the DREAM complex is assembled by a phosphorylation on human LIN52, and this phosphorylation site is induced by a specific kinase DYRK1A. DYRK1A has been studied in many other contexts, but we here ask then whether DYRK1A by assembling the DREAM complex could impact DNA repair gene expression. But DYRK1A, there are a range of molecules that inhibit this kinase. The most well-established is a natural compound harmine, and as an additional control, we used an independent chemical INDI that also inhibits DYRK1A. My hypothesis was that if we inhibit DYRK1A, we eliminate the phosphorylation, DREAM gets disassembled and thereby inhibited, and would lead to a de-repression of all these DNA repair genes. And this, we propose, could become a strategy to prevent mutation accumulation and therefore prevent cancer and promote longevity by keeping our genomes more stable. Let's first ask, does this work? So here we use now quiescent human cells because DREAM is assembled in G1 and G0. And we treated these quiescent human cells with either harmine or INDI, and we see a de-repression, an induction in either case of DREAM target genes, and many of them are in fact DNA repair genes. Does this inhibition by harmine or INDI confer resistance to DNA damage? This we show here. We treat these quiescent cells with UV or with alkylating lesions, different lesion types requiring different DNA repair mechanisms, and we see in both, as expected normally, a high apoptotic response, but that is significantly reversed by treatment with harmine or INDI, suggesting that they are, just like our worms, resistant to DNA damage. Could this be applied in vivo? Here, as a proof of concept, we turn to a very specific degenerative process, and that is retinal degeneration in humans associated with age-related macular degeneration. We know that these cocaine syndrome patients that I showed you, that has very early retinal degeneration, this can be recapitulated by a mouse that has the exact same mutation, but it takes long in this mouse, much longer than in the human. They then lose the outer nuclear layer, which is the layer on the retina that contains the photoreceptors. So it's sufficient to have a repair defect to accelerate the degeneration of retinas. Then we looked at humans that developed age-related macular degeneration, and we looked at the gene expression data of the retinas of those AMD patients compared to age-matched controls, and asked what kind of genes are downregulated in these patients, and we see that transcription-coupled nucleotide excision repair genes are downregulated, the same repair mechanisms that's mediated by CSB. Now, we use here a mouse model that is the most prevalent prodroid mouse model because there's most prodroid syndromes, ERCC1. It's a nucleotide excision repair gene that's knocked out and leads to very rapid degeneration and including retinal degeneration. And we treat these mice for 2 weeks with Harmine and then assess the photoreceptor apoptosis by tunnel. And this normally doesn't occur in wild type at that age, but in ERCC1 it occurs very early already. And this is significantly reduced here, the numbers, when we treat these animals with Harmine. And this experiment was done by Kalina and Georgina in George Carreras' labs, who collaborated on these mouse experiments. Then lastly, this is my last data slide, is the damage actually dampened by Harmine treatment. Here we see the buildup. You can see that because of the nuclear structure, particularly in the inner nuclear layer in the retina, you see a strong buildup of gamma H2AX because of the repair defect. And this is significantly dampened by Harmine treatment, indicating that they can repair these endogenously occurring DNA damage. What did I show you here? I showed you that the DREAM complex functions as master regulator of DNA repair capacities. In C. elegans, these DREAM mutants elevate the resistance to DNA damage by improving repair kinetics. The DREAM mutants de-repress somatic expression of DNA repair genes and confer germline-like repair capacities, because most of these DREAM target genes are usually only expressed in the germ cells. The DREAM complex inhibition in quiescent human cells elevates resistance to distinct DNA damage types. The DREAM inhibition by Harmine in vivo protects from retinal degeneration in ERCC1 prodroid mice. And we propose that the DREAM inhibition offers now a pharmacological route for enhanced genome maintenance to prevent DNA damage-driven cancer, both during development and with aging. And I acknowledge the people who did the work that I presented. Thank you very much for your attention. We'll be happy to take questions. And if you're interested in any of the work that I've presented, we are recruiting postdocs. Thank you very much.
Thank you very much. Was great. Very enlightening. So, for the first part, in the right part of the slide here, in the mutant in the male mice. So, in the first generation, these mice are fine, no? But they are completely destroyed at the same time. So they have the genome already damaged everywhere, yes, in all cells, yes, no, or not? But they're fine. How that's possible?
So that is really fascinating. We think that that has something to do with the post-mitotic structure of the adult worms, where there is sufficient time to transcribe genes. So the double-strand breaks that we induce, double-strand breaks are unlikely to directly interfere with transcription. So that is why we think they can still function relatively well. We indeed see no overt pathology. There's some slight delay in their development, but it's really not very strong. So they seem to be coping quite well with having fragmented genomes. And my explanation for that would be that fragmented genomes do not pose such a big problem on transcription and on quiescent or in post-mitotic cells. The problem is really what happens during mitosis. That's really the big challenge. And there, it's probably this breakage fusion bridge cycle that allows them to continuously divide. They lose some DNA, they gain some other fragments, but and maybe this has also to do with the chromatin structure changes that allow them to undergo cytokinesis despite having fragments in the DNA. There could be some help by the holocentric structure of C. elegans centromeres that could work in their favor. But the fact that we see the same fusions in humans suggests that this is actually also happening in humans.
And there is an aberrant expression overall in these cases, beyond the genomics. There is a complete alteration of the patterns there, or not?
Not a complete alterations. What is the most profound thing that you see in the gene expression is an overexpression of X-encoded genes in the daughters, and that is specifically because you lose the silencing of X in meiosis. So that is the most profound thing you see. Otherwise, there's not so much expression changes except these heterochromatin factors. I mean, by far the strongest induction of anything is this H1 linker. So it's really the heterochromatin that is changed predominantly.
Thank you. And I had another about the TMEG signature in humans that you see in aging humans. This also increase in cancer in humans?
So there is the most of the work in humans is indeed related to cancer, and there's a lot more coming out right at the moment because it's still a relatively new pathway. And it was initially thought that in cancer cells, it's the backup pathway that is used when there are defects in homologous recombination repair. And there could be also connections to PARP inhibition and things like that then TMEJ becomes very predominant. Our data suggests that it's actually very important, at least in germ cells, a very important repair mechanism. There's now new evidence that shows that even also in ES cells, in human ES cells, this is actually a predominantly acting repair system. And I think the more we look at this now, we will actually find out a lot more usage of TMEJ.
And the final one, second part. The DREAM complex in humans, do you remember the pattern of expression in tissues? What is the tissues that express the most and the least? A little bit.
So in humans, it's mostly about the regulation of its assembly. And this is very dynamically regulated. That in each S-phase entrance, it's disassembled because it also regulates a lot of cell cycle genes. That's what it's known for most of its life being studied. And so it's disassembled in every S-phase entrance and reassembled in every G1. So it's mostly the difference between cycling and differentiated cells. What we find actually particularly exciting to look at now, we've generated some new mouse mutants for DREAM mutants where we can specifically inactivate DREAMs. One thing we are very excited about is the hematopoietic stem cell that is mostly quiescent in its life, and it accumulates NHEJ products. I mean, these are the products that you see then being expanded in leukemias, for example, because they use inaccurate repair. And what we like to propose is, what happens now if we actually inhibit the DREAM and allow them to repair better? Would that prevent eventually the clonal expansion of these end joining products? So I think there in these cell types, it's particularly interesting to look at.
Thank you. Questions from the audience?
Björn, thank you for the talk. It's a beautiful, inspiring talk. And I have just a more general question regarding the first part. You based your studies on irradiation. So, but what about alcohol? That we are all exposed, so males and females. So, have you tested if aldehydes, alcohol drive also the transgenerational damage?
It's a very interesting question, and we should absolutely do that. Yeah, I mean the prediction is that any genotoxic influence, particularly in mature sperm, will have detrimental consequences because of this highly compacted structure. And it would, of course, be the question how exposed they would be, because obviously there's a lot of detoxification in the organism. But this is certainly a very, very important and interesting question to ask. We haven't done that experiment yet, but I think it's a great idea that we should do that.
And do you think that the DREAM would also rescue transgenerational damage? The DREAM complex, if it also applies for the first part?
We haven't directly tested that. However, the DREAM is not active in germ cells because it's disassembled there. So that's why all these targets are actually expressed in the germline and not repressed. We haven't tested it, but the prediction would probably not. But whether they mitigate some of the somatic genome instability in the daughters and sons, that is an interesting question. We haven't looked at that.
Hi, very nice talk. Yeah, thank you. I have a very simple question. I was wondering if the DREAM mutant worms, they live longer or no?
So they live longer under conditions where we drive aging by DNA damage, because naturally in laboratory conditions, DNA damage is not limiting their lifespan, very unlike human and mice. So therefore, they are not long-lived as such. Only under conditions of DNA damage they are long-lived compared to wild type.
Okay, and maybe something related. I mean, if you look at GWAS study in human, for instance, maybe you will find mutations that are associated to centenary or something like that.
Yeah, that's a very interesting question. And what we've looked at together with Yusin Su, we looked at whether we find variants that are related or enriched in human age-related diseases. And there we find a very strong enrichment of subunits of the genes that are coding the subunits of the dream. So this is very preliminary at this point. But I think this is a very exciting prospect that it could be really the level of DNA repair capacity that determines our susceptibility of age-related diseases.
Thank you.
Very nice. Regarding the second part, so it is well known, as you mentioned also, that the dream complex components control cell cycle have a very important role in the checkpoints. So can you exclude that all your observations, at least part of them, could be due to the regulation of the checkpoints, not necessarily directly to the DNA repair by itself?
Yeah. So this is in C. elegans very well characterized from the very first genetic experiment from Bob Horwitz. That is that mutations in the dream genes are insufficient to drive cell cycle. You need this, why they are classified originally as so-called syn-merf genes. This is synthetic multivulva. And not delving too much into C. elegans genetics, what that means is that there are multivulvas are formed when there are additional cell divisions during development. And this only occurs when you have synthetic mutations in other pathways, in other syn-merf pathways. So in itself, despite de-repressing cell cycle genes, it's insufficient in dream mutants to enter cell cycle. And these are in C. elegans, it's almost all post-mitotic tissues. So there are no extracellular divisions in these dream mutants. This property is very conserved in mammals, where a dream itself is insufficient to drive cells into cell cycle. You require an additional very strong oncogene. So for example, in the context of a CRAF, you would get additional proliferation. Or with retinoblastoma itself. Therefore, when we look at these quiescent cells, we have a very, very, very mild fraction of S phase cells that is elevated, but it's very mild. And that's why we also here started with photoreceptor cells, because they're post-mitotic. So that's why we also think it could be safe to inhibit dream because of its insufficiency, but you wouldn't want to do that in the context of a strong oncogene.
Thank you.
Hi, I'm Ollie. I work with Marcus, who's like the histone guy here. So since he's not here, I think I'm going to be bothering you instead. But I had a question about your H1 studies, where you looked at how H1 kind of inhibits the access. Have you ever looked into the mutations of H1 or anything like that?
So we looked in C. elegans, we then used the three most induced subunits of H1, and His24 we used here because it's viable. Other subunits of H1 are essential, so we couldn't really looked at that. So that's why we did here most of our experiments with His24, and then we backed that up with HPL1, but not with, we didn't study extensively other subunits of H1, because the challenge is to identify those H1 subunits that are not essential, because otherwise the worms would just be dead.
Hi, thank you very much for your talk. I really enjoyed it a lot, very educational. And I was just wondering, I mean, I'm going to ask you to be a psychic rather, because I just wonder what do you think about the potential application of this knowledge to the maybe to some potential clinical approaches to prevent gonadotoxic effects of chemotherapy in patients.
Yes.
So given that, I mean, I think that, so you explain us like these different mechanisms in terms of how to handle DNA damage in germline and in soma. So I don't know if you think that there would be like some way to really prevent or address somehow the gonadotoxicity.
Yeah, I think this is a really important question. And I think the first direct consequence of this work should be to much better define the genotoxicity in males of the chemotherapy. So there are clearly genotoxic consequences. And our data would suggest that the period of genotoxicity is very specific to the differentiation state of the during spermatogenesis. So that in the direct aftermath of a chemotherapy, you should abstain, but you could regain stable genomes by the newly generated ongoing gametogenesis. So I think the very first application would be to better define the window of abstinence, because the problem currently, one of the problems is that the suggested periods are extremely long, and men being men don't adhere to it, and so that could actually be then a problem, right? Because of course there's cryopreservation and all that before, but things could actually contribute quite dramatically to what we see now in this rise in autism and things like that. So I think there's a direct consequence, and that could be done immediately by now better defining the windows of genome instability. And you could actually do that. It's not very difficult to do. That would be the direct things. But of course, I think there's a lot of opportunity to think about protection. So also when I think about the dream, one thing that is very dramatic in gonadoprotection is actually the consequence of chemotherapy in females, where this often results in the complete destruction of the oocytes. And here, I think to have protective mechanisms, like for example, DREAM could be potentially such a protective mechanism to protect the oocytes from undergoing apoptosis, because they have a very low threshold for that. And so I think there are many translatable elements here that could really make quite a difference for chemotherapy patients.
I guess, I'm sorry, but I guess that there you could have like this, that maybe this kind of protection is impairing the effects of the chemotherapy. Like there could be like some interaction that maybe could be like, so do you think that there could be like some way to like go like target specifically like?
Yeah, so I mean there are certainly ways to depending on the tissue type, but to have targeted therapies, targeted applications, for example, of a DREAM inhibitor. I think that is very well possible, and you can also see whether there's a timed requirement, whether you could do it after your. I mean, cisplatin, for example. A problem with cisplatin is it stays around for decades in the human body, but you don't really want it for decades, right? I mean, you want it acutely. And I think here you could have a mitigation then afterwards. And I think it's particularly interesting nowadays that with all the survivors that we have from chemotherapy, that they actually show premature aging because they have in the aftermath all these consequences of genotoxic effects. And I think there's a direct application of improving DNA repair capacities to mitigate these long-term consequences of chemotherapy. So I think for that, it could really be highly beneficial.
Thank you very much.
All right, thank you.